in protein structural biology. So, of course, that's a question, why should you, or why any of us would be interested in protein structural biology? So the first thing is that, for example, you are selecting or identifying genes or proteins that you are interested in, or if you really want to find proteins or genes that, for example, on the surface, because you are developing a vaccine, or a protein that is responsible for a specific metabolic pathway, then you may have to use structural biology to identify the protein and to find out those protein regions that are particularly interesting for you. The second thing is that if you have a change in the amino acid sequence, for example, you are looking for the same genes, the homologous genes in different species, and you find out that there are some SNPs, some amino acid changes between two proteins of the same function, and you are looking for the effect of that change. This is especially true if you are, for example, looking for disease-causing mutations in humans. So that's one of the main reasons why you are going into the protein structural biology, because you try to identify or investigate the effect of that particular amino acid change within the protein. The third one, which is, I think, the third one which should be relevant for you, is if you want to find potential target sites on proteins. Target site means here can mean two things, I think, especially in our world. One is if you're looking for epitopes. So again, for example, if you look for a potential vaccine target, then you will really need the structural biology to find the potential sites. And the other thing is that if you are thinking about targeting a protein with a drug, a small molecule, to inhibit that particular protein or its function, then again, you have to identify that site by using structural biology. Of course, there are a lot of other things that you can do. You can investigate the evolution of the structure-function relationship. So if you look at the same domain, the same enzyme in different parts of the phylogeny, then you can find out how it changed, what are those parts that are never changing, what are the most conserved bases, and what are those parts that maybe gave a new function or an altered function for a protein. Also, there is a complete area in protein structural biology which is just focusing on folding. So you will see that most of the proteins, especially in prokaryotes, they have a tertiary structure, so they are forming a well-defined three-dimensional structure in their native state. And understanding how this is happening is actually a huge challenge even nowadays. So, and this is what the folding is about, how that protein, is reaching the final three-dimensional structure, which is the stable, the functional structure. There is also another area which is now, even I think it's not just simple, but it's a very complex area nowadays. You can do high throughput testing on target sites, and in this case the target site is much more about an enzyme active center, for example. So if you find out that in a cancer, for example, there is a kinase which is responsible for the cell proliferation, and you crystallize that kinase domain, you have the three-dimensional structure, then you will be able to screen small molecules, try to put small molecules into that reactive or active center, which is responsible for example, the phosphorylation of another protein that will lead to the proliferation of the cell. This is always in. So there are, different steps how you can screen your small molecules. There is, of course, an in vitro step when you are doing the whole screening in 69, 96 well plates or even bigger plates. But usually you are starting with a library of small molecules of hundreds of thousands. And usually the first step to shrink this data set to have a smaller set of target or potential molecules is an in silico step when you try to put your molecules on the protein. And of course, there are many other ways you can use protein structural biology. So the first thing what you have to understand, because anything you do in silico, anything you do with bioinformatics is always based on something that was done in vitro. If you just visualize a structure, you are working with a file which was created by someone who crystallized that protein. If you use a predictor, for example, if you want to predict a secondary structure, then you are using a program that was trained on structures that were made in vitro. So in the protein structural biology world, everything, really everything is somehow connected to a previous in vitro experiment. If you think about how you can investigate how you can identify the structure of proteins in vitro, then there are two main categories. You can make a high resolution structure, which will be also, it's a bit more local structure because you are looking at usually only a segment or just one domain. It's very rare that you can identify the full structure of a multi-domain protein, for example. There are cases, there are crystallized ribosomes as well, but that's something what you will get a Nobel Prize for. So that's not really common. It's much more common that someone is expressing a domain. If you are lucky, then your protein is only composed of one domain, but in many cases they have multiple domains, and you can express one domain, you can crystallize one domain, and then you can identify the structure. And if we speak about high resolution structure, we are mainly speaking about two different in vitro methods, the Röntgen crystallography, X-ray crystallography, and nuclear magnetic resonance. The X-ray is the old and gold standard way of identifying structures, but you have to know here that to make a good crystal, you need a very high concentration from the protein. You usually have a very high concentration of salt, so it's very far from the natural environment, and also this is completely static, so you don't really see the dynamics of that domain or the structure. What you have is actually a snapshot of that structure, and during the crystallization, there are many possibilities to introduce a bias or introduce some kind of artifact, especially because of the high protein and high salt concentration. The other one is the NMR, so the nuclear magnetic resonance, which is giving you a dynamic, much more dynamic picture, so you can see movements also. This is a very important part of NMR, actually. You can easily use a completely natural environment, so sometimes they are just simply use the normal pH and normal salt concentration, but even sometimes even nowadays you can do in cell NMR, so you can overexpress your protein of interest inside a cell, and then it will be completely within the natural environment. They are usually doing this in E. coli, of course. The downside is that it's not really deterministic, so you never have one structure. If you search for an NMR structure and you download the file which is containing the structure, in case of the X-ray, you are downloading one structure, so you will have a coordinate for each atom once in your file. If you are downloading an NMR structure, the file will usually contain 10, 100, or 1,000 structures. So what is happening is that there is a post-processing after the NMR analysis, and a computer program pipeline will create an ensemble, will create 100 or 1,000 structures that are describing the observations the best. There is no gold rule which one to choose, so that's actually a problem. You know, I cannot tell you that from the 100 structure you should use the first one, or you should use the 21st one, or you should make an average. Never do an average. The best way is to plan your in silico pipeline by we have some microphone activated here.
No.
Not anymore?
Not anymore.
Okay, thank you. So, so the best way to work with NMR data is, if you want to do something with that structure in silico, is to work actually with more than one from this 100. So try to work with all of them, or try to work with 10 as much as you can. A very big downsize or con for the NMR is the size limit. So as I told you, with the X-ray, even a ribosome can be crystallized, but crystallizing a few hundred amino acid domain is perfectly okay. It's not a huge challenge. And the NMR size limit is somewhere around 150, 250. So you cannot really go above that amount of amino acids. So if you have a larger domain, you won't really be able to identify the structure with NMR. The other way to investigate the structure is to do a global structure. So you don't want to go down to the atomic resolution level. You don't want to show all the side chains of all residues. You just want to have a picture of the shape of your protein. One of the oldest method is actually a simplified method of the X-ray. So this is the small angle X-ray scattering method, which will give you much more like a shape. The numbers here are always showing you that what kind of structural level you can identify. So this is the secondary and the tertiary structure here. With these methods, you are much more looking for the tertiary or the quaternary structure. So it's these methods are extremely good for making a better picture on protein complexes. So you will see how the different parts of protein complexes are assembled. So that's one of the old ones. I think the trendy method at the moment is the cryo-EM, when you just slice cells usually and try to identify your protein of interest with a very high resolution electron microscopy. This is something which always helps. So what you are doing is that you try to label your protein, you try to find your protein in the huge mixture of your cytoplasm, and you try to identify it as many times as you can. So usually what you do here is you have hundreds or thousands of slides, and you try to find your protein in all of them, and you will have transections of your, or like a cut, different cut from your protein from very different directions, and you try to build up your final shape from these different sections. Also, a quite often used method is this circular dichroism. I think it is pronounced like this, where you basically use the different light scattering properties of the alpha and alpha helix and beta sheets. So this is not a good method to identify the whole, all the different parts of a protein structure, but it is good if you want to have a picture about the alpha helix content, for example. So you know that your domain or your protein has a certain amount of alpha helix in it, and you think that, for example, due to or during an interaction of another protein, or during just heating up, this alpha helix content will be changed, and this is what you usually visualize or analyze with the CD measurement. It's very good because it's dynamic, but it gives you a very low resolution picture. And there are some other old ones, like the fluorescence spectroscopy, when we utilize the inherent fluorescence of the tryptophan and the tyrosine. They are emitting different wavelengths, now different intensity if they are exposed or if they are not exposed. So both of these amino acids are highly hydrophobic. They are usually sitting inside the protein, somewhere in the middle, so they are not exposed. And what you can do is when you heat up your protein and look for the unfolding of the protein, these big hydrophobic side chains will be exposed, and you will detect the fluorescence pattern or fluorescence signal. And also there is a very new thing, which is one of the big promises of structural biology, I think, for the next 10 years maybe, which is the linear laser. So it's actually a big laser beam that is transformed to an X-ray with linear magnets. And what is happening is that instead of having a constant X-ray source like you have in the X-ray in a normal crystallography, here you have pulses of extremely high intensity radiation. They are actually evaporating instantly what is in front of them. But before the evaporation, the protein is scattering these radiations, the X-ray radiations, and you can detect that scatter. The special thing with this one is that it's a bit like the cryo-EM. So one evaporation, one photo of a protein is only like a dissection. But again, what you do here is that you are using like thousands of pulses per second, and you are evaporating thousands of proteins, and you will always have a different scatter graph, and from this you have to assemble your final structure. This is not, I don't think it's working yet. I think these were used for virus particles already, so you can see very big structures, but not really a high resolution structure. So how would you go forward with the in silico investigation? Of course, again, what we have seen here, the primary, the secondary, and the tertiary structure. The primary structure, we spoke about this already a few times. So, of course, if you just have the amino acid sequence, you can do multiple alignments. You can use BLAST and find homologs look at conservation of different parts of your protein. That will give you already a lot of information. You know, if you, for example, if we go back to that example when you are looking for a disease-causing mutation, so you have a mutation that is that you found, and you think that it is causing some special disease in animal or humans, then you can just look for all the homologs of that special protein and look at that position which is mutated in yours. And if it's extremely conserved, if you see the same amino acid that you have in your Y type down to Drosophila or even down to prokaryotes, then you would have a good chance that changing that amino acid will really make a huge change in your protein. Of course, you cannot really say at this point if it will alter the structure, if it altered the function, or if it altered the function through the structure. So that's not enough. You just this is not enough for that to answering that question, but you already have a clue that this is probably a significant change. I cannot emphasize enough that the substitution matrix is an extremely important thing here. All the substitution matrices we have, the substitution matrix gives you an idea of the significance of a change. So obviously, if you change a big hydrophobic side chain to a small hydrophilic one, that's a huge change. But it's not always the same in every proteins. So we have very good substitution matrices for globular proteins. I will tell you soon about that. But there are problems with transmembrane proteins and disorder proteins, and again, I will speak about them in a few slides. So the secondary structure, so determining if special or parts of your primary sequence are forming alpha helices, beta sheets, or just simply loops. First of all, you can just simply visualize your structure. So if you are lucky and your protein of interest is already crystallized and you can find it in Pdb, I will tell you how to look for these proteins. Then you are just simply download the file, visualize, and it will be visible already if it's an alpha helix or a beta sheet. If you are not that lucky, then you can predict these secondary structure elements. It can be extremely important. I will speak about this later, why the secondary structure is extremely important. It's not just because these secondary structure elements are the building blocks of the tertiary, the complete structure, but in many other cases, secondary structures have other significance as well. So you can predict from the sequence. They are not extremely reliable, but you can try. And you can also use molecular dynamic simulations to check if your protein wants to go into an alpha helix or beta sheet. And finally, the tertiary structure, so visualizing the whole domain or whole protein and its whole structure. Again, you can just simply visualize if you are lucky, and you can download it from Pdb. If you have the structure, you can also measure global parameters, especially, so these global parameters are usually measured or calculated or analyzed when you are doing some kind of molecular dynamic simulation. So when you're not just looking at your static molecule, but you want to compare it with someone. So if you, for example, compare two domains from two different organisms, or if you are comparing your Y type and mutated domain, then you can check RMSD and RG. RMSD is it's a kind of average square root distance between the atoms. So if you think about those figures, I think many of you already saw like superimposed structures when you have two different structures, but very similar, but they have some kind of difference between them. So what is happening during the RMSD calculation, that all atoms, or all alpha carbon atoms usually, are compared to each other in the two different structure, and an average distance between these will be calculated. So this RMSD, of course, increases dramatically if you, for example, starts unfolding a protein. So if you heat up a protein and it starting losing its globular structure, it's almost like starting exploding, then this RMSD will increase dramatically, because all the atoms will be far away from their original place or original environment. The RG is way much more simple. That's the radius of gyration. So that's actually just giving you a number of the two most distant part of your protein. So again, if your protein starts exploding because of an unfolding, then it will occupy more space. So the RG is just actually a parameter on what's the amount of space the protein is occupying. If you don't have your structure in the PDB, but you are lucky, a bit less lucky, but even lucky, then you have homologous domains, homologous proteins in the database. So you don't have to have your own protein, but if you have one, or most or much better if you have more than one, like 10 or 20 or even hundreds of structures from the same domain, you can just simply download them and use homology modeling. It's a very quick way to create your own structure. So it will use all the structures that is available. It will use the sequences, make a sequence alignment, and based on the sequence alignment, it will create the program, the bioinformatics software will create a homology model for you, which is actually a very reliable way to make your three-dimensional structure. For the tertiary structure, you are very often doing molecular dynamic simulations. I will speak about molecular dynamic simulations later, so just don't worry about that now. You can do folding, unfolding. This is again a very unreliable part of the in silico tertiary structure analysis. I wouldn't say that it's very trivial to do folding. The big problem with folding is, so let's just imagine that you are starting with your protein sequence completely stretched. So it's like a string. You have 150 amino acids right next to each other, and you try to decide from that what is the folded structure of the same sequence. What's the best folded structure? The problem is that the folding funnel actually is not smooth. So if you have something, if you have something like this on this picture here, so the left-hand side, if you would have something like this, and this will show you the structural space. So at the bottom, you have the lowest energy and the ideal structure. And here you have all the high energy structural variants, for example, the completely stretched proteins. So as you can see, if you start from here, and you just always go for a lower energy state. So anything you do is just look for what is the change I can make on this structure to make the energy level smaller. Then you will actually fall into the energy minimum. But folding energy surface never looks like this. It always looks like this, much more. And what you can see here, or even you can see here on the 2D graph, if you start going down, but you are here, then then from this point, anything you do will increase your energy state. So you may think that, okay, I found the energy minimum, but it's actually very far from the energy minimum. And there are a lot of like intermediate holes here. And this is actually not, it's not an artifact. That's normally how it goes. That's why cells actually need chaperone proteins. The chaperones are usually help during the folding as well, and help the proteins to achieve their final folding state. During the real folding, many proteins will stuck here or there, and they need a lot of, they just need a small energy to push them out from this hole and try other energy minimums as well. So that's why simulating a folding is actually a very challenging task, but I will tell you how you can do that. It's a bit easier to simulate in molecular dynamics simulations the unfolding, because you just have to increase the temperature and you will see how the protein unfolds. But the problem again, that it can unfold this way, that way, and many different ways, and maybe you won't see that much of difference sometimes, or sometimes you will see a dramatic difference. So if you want to do unfolding, maybe the best way is to do it like 100 times and then try to build up some kind of unfolding funnel from that. Clearly, you won't be able to identify these spots, because if you think about the unfolding, so you are starting from here, from the bottom, and you are giving energy to your structure. So it will always project, it will always go up. So you will never see these intermediate states. And these intermediate states are sometimes very important. So simulating unfolding is not the same thing as looking at the folding, unluckily. But there is a way we can deal with this, and this I will also speak about. This is the replica exchange molecular dynamics simulation. Okay, so before we go into the methods that we can use, and the databases we can use, and the data sets and the data formats, we have to speak about the three main categories, because this actually constrain what can we use, what kind of data sets, databases, methods, what should we actually see as a protein structure. So if you look at the eukaryotic genome, we have about 1/3 or eukaryotic proteome actually, 1/3 of the proteins or protein regions are globular, 1/3 is transmembrane, and 1/3 is disordered. Globular means that you have a well-defined three-dimensional structure with a hydrophobic core and a hydrophilic surface. The whole structure is stabilized by the hydrophobic core, and everything that is happening on the surface, or small packets on the surface maybe. The main protein functions, of course, almost all the enzymes are globular, all the catalytic domains, and there are many interaction domains as well. Why a catalytic domain or an enzyme has to be globular? Because for a catalytic center, we almost always need a few amino acid side chains, 3 or 4 amino acid side chains that are going into that active center or packet, and they have to be oriented in a very special way. So you actually need a scaffold, you need a frame to hold these amino acid side chains. That's the whole idea and the whole reason why you have a big globular domain. Sometimes you have 200 amino acids just to create a frame for that special 4 side chains that will do the catalytic activity. So kind of not really how would you say, it's not, it's a bit of spare of amino acids, but this is how it works. If you think about transmembrane proteins, which is again another 1/3, these two categories a bit more overrepresented in prokaryotes, and this order a bit underrepresented, but it's not completely general. As soon as you have a bigger genome, you will have more disordered proteins. I will show you a picture because it's a very interesting one actually. How the disordered proteins are exist in prokaryotes, but but now speak about transmembrane proteins. Of course, here we have all the receptors, all the transporters, channels, ion channels, water channels, and also we have here the first steps of cellular signaling. So if you think about the signal that is coming from the outside, the transmembrane proteins are the first element of this signaling chain. Extremely important proteins, very, very important in many ways. As far as I remember, like 80% of the human drugs are targeting transmembrane proteins. So if you think about any antidepressant or even cardiac drugs, they're all targeting some kind of transmembrane receptor, channel, or proteins. Extremely important again, as I said. And the third category are the disordered proteins. So the main definition for protein disorder is that these are proteins or protein regions that doesn't have any well-defined three-dimensional structure. So when you think about the traditional way of thinking about proteins, that you have a DNA sequence and then you have, which is determining the amino acid sequence, and the amino acid sequence is determining the structure. This is the old paradigm. This is not working for these other proteins. They are very flexible. They can have multiple different structures. They can use different structures to interact with different protein partners, and this is their main advantage, and this is why they are usually highly overrepresented in anything which is based on protein-protein interaction. So if you think about transcriptional regulation, if you think about signal transduction, this is much more in eukaryotes, but also a little bit in prokaryotes. But also if you think about connecting two proteins to each other or multiple proteins to each other, we usually use the adapter function if you connect two proteins to each other, and we use the scaffold if you connect multiple proteins together. So if you think about the ribosome is composed of RNA and also a few small globular proteins, and they have to be glued together. And the glue is actually this disordered protein, which is just go around and interact with different proteins. But if you also think about long distance interactions, this is very important. In many cases, when you have to reach a distant part, maybe the best example is the endocytosis. So when you have to fuse a membrane vesicle, those two sides, they have to come together, and actually these long distances are usually bridged by these ordered proteins as well. The question is, so this is just the basic categories and basic functions. But if you are coming from the other side, so if you are not coming from the structural biology side, but you are looking for a specific function, and I try to choose those functions which are actually relevant for all of us, or maybe in the prokaryotic world as well, then you have to prepare for these kind of protein structures to deal with. The easiest way is the metabolic pathway. So if you look for any glycolysis enzymes or beta-lactamase. So if something is, cut another molecule, which is usually a catalytic domain, then you will usually end up with a globular structure. In most of the cases, you will find some kind of homologue in the database, so you will have a very good chance to get a good structural information. They are also soluble, so even if you don't have a structure, it's not extremely hard to identify the structure by just crystallizing or by using NMR. And also, they are almost always, of course, intracellular. If you are dealing with cellular signaling, then you will have some transmembrane proteins. Sorry for these abbreviations. I will always tell you what is that. TM is for transmembrane. Because those are the first steps, but you will also have a lot of globular and disordered complexes. In many cases, if there is a chain of cellular signaling, then for one globular domain, there is a disordered partner. The best examples are the phosphorylation chains. So if you have a kinase domain, which is globular, it will phosphorylate usually a disordered region on a different protein. And of course, you have to know, if you are dealing with a transmembrane protein, which part is extracellular and which part is intracellular. Of course, because the extracellular part will be responsible for taking the signal, and the intercellular part will be responsible for giving the signal forward. If we are speaking about protein complexes, so your main target is to find out, for example, how the flagellar protein complex is built up. What are the most sensitive parts in that protein complex? You want to, for example, attack. Then you have to deal with globular proteins, and with 100% sure there will be some disordered proteins as well. If you are going for transcriptional translational regulation, again, usually a globular protein with a disordered partner. This is very similar to the cellular signaling. If you look for cell surface epitopes, so again, if you think about developing vaccines, for example, then you have to deal with transmembrane proteins, and the most important thing you have to focus on is the extracellular part of that transmembrane protein. So this looks very obvious, of course, but if you don't know the protein, if you don't know anything about the structure, then you have to decide if it's a transmembrane protein or not. If it is a transmembrane protein, then you have to find where is the transmembrane domain. Then you have to find which one is the extracellular part. So it's not extremely obvious always. If you think about molecule export-import, so for example, you are dealing with drug pumps, then you again encounter with transmembrane proteins, sometimes globular. The globular part here is sometimes extremely important because many of these export-import mechanisms in the membrane, and this is also eukaryotic, prokaryotic membrane, so this is not eukaryotic specific, they are active transporters. So even if you don't know how to identify the structure of the TM part, which is usually way much harder, the transmembrane part, which is responsible for making the pore or bringing the molecule through the hydrophobic membrane. But if this is an active transporter, then there will be a globular part attached to the transmembrane domain, and the globular part will be probably an ATPase domain. So it will use energy to help the active transport. And these domains are way much better known. So even if you don't know the transmembrane part of the protein, but you understand how the energy creating mechanism is happening in the protein, then you just enough to inhibit the globular part. The transmembrane part will not work without the energy. And again, transcriptional modification, extremely important, not just in eukaryotes or prokaryotes as well. there is usually a globular domain which is doing the modification, and there is usually a disordered region which is getting the modification. We are speaking about glycosylation, phosphorylation, ubiquitination, any kind of post-transcriptional modification. You need a well accessible protein region to put the modification on, and this is usually a disordered region. So again, what the main take-home message from this is that try to always look at the mechanism first, how that particular target is working, what is happening with that protein, and then it's much easier to decide how to go forward. So how you can find these proteins? If you have the sequence, you can use, of course, BLAST to find other. So let's just think about in the first line that you have a protein sequence that you don't know anything about. If you BLAST, and if it's a globular protein, then there's a very good chance that you will find homologs, and you will find out the function. You may find out the structure very easily. If you have a transmembrane protein or a disordered protein, you have a much lower chance to find homologs. For a human protein, a human enzyme in the glycolysis, I think, it's very easy to find even a prokaryotic homolog. But anything from Drosophila to the mammals, they will be that conserved that you will find. For transmembrane or disordered proteins, it's even sometimes hard to find a good homolog within vertebrates. If you are lucky, there will be a few in other mammals, but it's very rare that you find, for example, a homolog for a human transmembrane protein in fish. There should be a homologous functional protein, but in terms of sequence, they will be very different. And here again, we have extremely good substitution matrices for BLAST and for globular proteins, and we have very bad substitution matrices for transmembrane and disordered proteins. The PFAM, which we already discussed a few meetings ago, the PFAM is very good identifying globular domains, again, because PFAM is based on the conservation of sequences. And if they are well conserved, then PFAM will identify them with a very high accuracy and reliability. You will get prediction in PFAM, transmembrane and disordered regions as well, but you have to know that if you see, okay, this is a transmembrane region, this is a disordered region, they are all computer predictions, and they are not extreme. They are way much less reliable than the globular prediction. Based on short motifs, it's very good to use short motifs in disordered regions. Short motifs, for example, a recognition motif for a kinase, a recognition motif for a glycosylation enzyme. If you have them and you know that your protein is disordered, then it's very good to look for these short linear motifs. The physical-chemical properties are very, very important. The globular proteins have a very average amino acid composition, so you cannot really use them. There are many hydrophobic residues. You know, they are needed for the hydrophobic core, but there are also many hydrophilic residues which are sitting on the surface. So just from the sequence, just from the physical-chemical properties of the amino acids, you won't be able to say that, okay, this is a globular protein or this is X, Y, or Z domain. It's much better to use these kind of properties for transmembrane and disordered regions. Transmembrane regions are always very hydrophobic because they have to be stable and they have to sit in the hydrophobic part of the membrane. And on contrary, disordered regions are usually very hydrophilic. So they are usually way much less hydrophobic residues than an average globular protein. disorder predictors can work for all of them. So why it is only designed to predict disorder, it will show, let me just show you something. I think I have the kit here. This is the kit receptor from human. If I just put the FOSTA sequence into IUPRED, which is not open, I think, yet. I will open it. Oh, not like this. So this IUPRED 2A is one of my favorite protein disorder predictors. If you just paste the sequence here, then, I mean, there isn't too many disordered regions, but these two parts here probably are disordered because the red line is go above 0.5. But what you can see here is that probably this one is a globular domain because it is below the middle line, but not extremely. And what you see here, for example, in this small region, it's a very highly hydrophobic region, which is completely the opposite of disorder. This is probably a transmembrane region. And if you go back to the structure and you go back to the Uniprot, then you will actually find out that, yes, this protein has one transmembrane region, which is here between 525 and 545. So it is the same part as the very hydrophobic one. I wouldn't say that you have to use this. I will tell you much better methods to find the exact location of transmembrane domains or transmembrane regions. But for a first look, as we did before with Ibrahim's proteins, this is a good way to have a good idea on the global structure of the protein. Of course, there are transmembrane predictors, and I will show you a few. Actually, I can show you now. One of the one I really like is CC-top. I will speak about this later, so I don't speak now, but the first thing, what you have to find out if it is really a transmembrane protein or not. Because if you just put in a globular protein into a TM predictor, it can easily be predicted to a transmembrane protein, or there are many different programs that will find transmembrane regions. What you see here, this is the same c-KIT protein we saw with the IUPRED, and the yellow part is the transmembrane region. And CC-top is actually predicting the topology well. There is a short signal peptide in the beginning, that's the small gray part. There is an intercellular part with blue, transmembrane region 1 in the middle, and then a red extracellular part. But these are here, these are all other different transmembrane predictors. They all predict the same transmembrane region, so that's okay. But there are multiple predictors that say that, okay, this is a transmembrane region as well, or this is a transmembrane region. We know that it's not. We know the structure of this part. We know, well, this is a soluble globular structure. And this is just a misprediction. So this is how reliable the transmembrane prediction is. I think the CC-top and the HMM-top is actually one of the best ones, not because I work with this guy, but also I know that this is really reliable. So transmembrane predictors are good, of course, for transmembrane regions. They can't be used for anything else. The next thing is Pdb. That's the protein data bank for structures. So if you look for Google for Pdb, then for me, that's the first hit, the RCSB Pdb homepage. This is the database that is responsible for storing all the three-dimensional structures of different proteins, and you can search with any kind of keyword. So if you want to look for the KIT protein, the c-KIT, of course, that's not that. c-KIT protein, then this is the kinase product complex of it. This is more than just one protein, of course, but what you have to know here is that the PDB is using a 4 mixed letter and digit identifier. So this will be your identifier for this protein structure. It's important because you can use this to easily download into programs. I will show you. But you also have all the other information here about, you can also look at the three-dimensional structure just here on site. So you can move your structure and look at the helices and look at different regions. You can also highlight, I think, regions here, but it's not as nice as working with structures in some specific programs, and I will show you soon. I never used the PDB. That's why I don't know how to use it. But if we just copy the ID, there are a lot of other fields here, of course, how this was identified. This is actually, I think, an X-ray structure. Where is it? Yes, in the method, it is an X-ray diffraction, and the resolution is 2.9 angstroms, which is not the best, but not bad. But if you just copy this from here, then I will show you soon what are those programs that can be used to visualize the structures. So PDB, again, you will find a lot of globular structures in PDB. You will find very few transmembrane domains. It's very hard to make crystal structure or NMR structure from transmembrane proteins. The main problem with the crystallization is that you have to use a very high, again, as I said, you are using a very high salt concentration, so the whole environment is extremely polar, and the transmembrane proteins are extremely hydrophobic, so they don't like that environment. There are some special ways how you can crystallize, but they are extremely underrepresented in PDB. You can find a few disordered proteins, which sounds maybe a bit strange, because I said that the main definition for disordered proteins that they don't have a structure. And how can you have a structure in the structure database? The reason is that there are complexes. There are quite a lot of complexes in PDB when you have the globular domain and you have a small part of your disordered protein in complex with the globular domain. And you can use these structures, of course, in your work. The Uniprot is the next one, where you can find quite a lot of information. So if you just go back to Kit here, then you can already have a lot of structural info just in the Uniprot. So you can see that where is the transmembrane domain. You can see which one is the extracellular, which one is the intercellular or a cytoplasmic domain. Again, this is important if you want to design an antibody against the extracellular domain, then you have to know which part is the extracellular domain of the protein, of course. But also have other informations here. Usually above the sequence line part, you have some secondary structure information here, as this one here. So here the greens are beta strands, the blues are alpha helices, and this one is the transmembrane domain in the middle, actually. And you also have other structural informations, like what kind of domains you have and where. So these are, again, globular domains you can work with. So Uniprot is also a very good source for having some basic information on your protein, especially if it's a known protein. But also I have to point here, it's very important that Uniprot is using predictions many times as well. So don't always believe everything what Uniprot says. Usually they say here, so if it's if it's a well-known structure or if it's just a prediction, then you will see. And if it's something like a prediction, then you have to handle it like that. And finally, there is a database for disordered proteins as well, which is called Disprot. Let me just open it instead of the PDB. This is the database. So this is dedicated to disordered regions. You can find different proteins. Of course, one of the best example for disordered protein is P53, where you can see which parts are disordered. So you can see that not the whole protein is disordered, but these ones, the green parts, are disordered. And you can see if this is known because it is, I think somewhere down here you have a lot of evidence on this, if this was identified by some kind of structural investigation or just by prediction. It's actually quite a lot here. Okay, there are different transmembrane databases as well. I will mention them soon. Okay, so if you know which category your protein is in, then let's see what is the level of structural understanding you have a chance to achieve. Again, if you have a globular protein, then you probably will be able to go down to all atom resolution. So you will be able to see every atom in your molecule, in your protein, where they are, all the side chains, all the bonds, hydrogen bonds, hydrophobic interactions, everything. Even if you don't have an existing structure, you have a very good chance that you can make a homology modeling and build up your own structure. Usually, what you end up even is a static structure. So even if you download it from the PDB, even if you make homology modeling, what you get is a three-dimensional model, a static model. So, of course, this is sometimes not enough. You may have to think about going further, but it's a good starting point, for example, for molecular dynamic simulations. And the first thing you will already see is that how your hydrophobic core is built up and how hydrophilic the surface is. You can always check these kind of parameters with very simple programs. If I won't list all these programs now because we would be here until like 8 p.m. But if you have a specific interest in this one, how can you screen globular domains very quickly for hydrophilicity or hydrophobicity, then I can give you a few tips. It can be very interesting because one of the main problems with a wrongly folded protein, for example. So even if you make a crystallography, sometimes the fold is not the real fold. I mean, the structure is not the real structure of the protein. And one thing you can be suspicious is that if you see a bit more hydrophobicity on the surface, as soon as you have hydrophobicity on the surface of a protein, it will be very unstable inside the cell. Hydrophobicity will be prone to aggregate the protein. Hydrophobicity is a signal for many intracellular protein degradation pathways. So if you have a structure and you see a big patch of hydrophobicity, then you may think about recreating the structure or repairing it. So if your protein is in the transmembrane category, then as I said, it's very rarely you will have any tertiary structure known in the PDB. but you can have a very good understanding on the extracellular intercellular parts. And this is when I say that even if you are working with transmembrane proteins, you don't have to be very sad, because there are many parts that should be known already. If there are not, big domains outside, inside, they are probably, as they are globular proteins, globular domains, they have probably structure known already. If this is not the situation, but if you have a much more very transmembrane protein, let me show you two examples of. The one is let's call the kit, the C kit, as one extremity, when we have, as you saw before, when we have a protein like almost 1,000 amino acids, and we only have one short 20, 30 amino acid transmembrane region. If you have this protein, you will know a lot about this protein, just because we know a lot about the extra and intercellular parts. Let me show you something else. This is TopTP. This is a topology data bank from transmembrane proteins. Again, someone may find out my name here, but this is not the reason why I wanted to show this to you. But if you look for example, multi-drug resistance proteins, just say MDR here, and even look for a Vibrio MDR protein. What you see here, this is generated like on live. What you see here, that most of the protein is inside the cell, inside the membrane, sorry. So you don't have those huge, easily identifiable extra and intercellular parts. You have a lot of things inside the membrane. You have loops. So if you want to target this protein with an antibody, then probably you can find some extracellular or intercellular parts that you can target. But in this case, it is not as easy to find out what to do. If you have something like this, then the first level you have to find out is a topology. So the topology is what you see here. This is the topology. This is, of course, not a nice, it doesn't look like this in the cell. It's not stretched within the membrane like this. This is just a topology. Which part is intracellular, which part is intramembrane, which part is extracellular, and so on. Together with understanding the topology, you also have to understand the different sites. It's not as easy, especially it's a bit easier in prokaryotic species, but it's a bit harder to understand in eukaryotes, because in eukaryotes, and it's also, I think, here in the document section, in different membranes. So if you think about the eukaryotic cell, there are so many different organelles there. So if you just think about the chloroplast or the mitochondria here, in mitochondria you have an outer membrane, an inner membrane, you have an intermembrane space. So defining the inside and outside is not always that trivial. In prokaryotic species, it's a bit easier, because there, of course, you have this double-layered membrane and cell wall, so that's not as easy there as well. But this is very important, because you have to know where your parts are if you want to access, or if you want to reach them with some kind of antibody or a small molecule. But this is the first thing what you want to find out. The next thing, and yes, for that I would use the CC top, which is a meta server, or I would use the HMM top alone. The CC top is building up a consensus topology, as you can see here, just based on the sequence. Consensus based on many different predictors. But HMM top is actually also a good standalone predictor, and much faster than CC top to have a rough topology. This one is also predicting the sites. I don't know if it is actually good or not. I don't know if it's not extremely user-friendly. I'm just thinking about if we have any outside extra side of solid. Yeah, so here we have some information about the orientation as well. It says that the blue part is outside. So if you know that this is a human protein, then the outside is always the outside of the cell, and the red part is the intercellular. So it's actually much more like this. If you think about the cell at the bottom and the outside at the top. Right. So that's the second thing you have to find out. And as soon as you have this, you may progress forward with your biological question. If you have a disordered protein, then the first thing you will try to understand is the secondary structure propensity. I will speak about this later, why this is extremely important. Because, as I said, the disordered proteins doesn't have any well-defined tertiary structure, but that doesn't mean that they don't have secondary structure. They can have secondary structure elements. They are not always stable, but this is again something which is an important property of these proteins. And I will speak about this in a few minutes. And also very important tool in disordered proteins to find out local physicochemical properties. So if you see a region that is a bit more negatively charged, a bit more positively charged, that will give you a clue that maybe that region, if it's, for example, if it's a region with a lot of positively charged residues, then you may think that that this protein is interacting with nucleic acids. That's quite often the situation, that if you have together with some kind of linear motifs as well, you have an enrichment of arginines and lysines, then it is potentially an interaction surface with nucleic acid. Or if you see hydrophobicity, as I told you, in disordered proteins, we have a very low hydrophobicity, but sometimes within a long, like a 50 amino acid region, you see two big hydrophobic side chains rise very close to each other. That's again a sign for something there, that maybe that small region is important for a reason. I will show you a region like this. And if you do NMR on these other proteins, you can do, or if you do molecular dynamic simulations, then you can end up with a structural ensemble. This is very, very similar to what we saw with NMR structures. So instead of having one three-dimensional structure, you will have thousands. In NMR, usually have 100. In these other proteins, you most often have 1,000 or 10,000 structures. That represents the most probable structural states of that disordered region. And then you have to continue your analysis with all these structural states. Okay, and I only have a few slides left. The first thing is, now it's much more practical. So you will see some programs here. So if you analyze the globular structures, the first thing you need a Pdb file. If you can download from the Pdb, you download it. If you have to make your Pdb file with homology modeling, then you will make it. For homology modeling, there are very good websites. If you are interested in this one, just drop me an email. We did a few homology modelings recently with Thomas, so I can show you a few very good websites to make homology modeling. But of course, the first thing you want to do is to visualize it. There are many different programs. Visualize on websites, visualize on your own computer. These are just a few of these programs. I almost always using Chimera. I think I can show you how it works. I hope. I actually had Chimera. Maybe it's here. This is Chimera. It will load in. Yeah, so if you want to download, it's the UCSF Chimera. That's what you have to search for, UCSF Chimera. I think it's a very user-friendly program. It's absolutely cross-platform, so you can use it on Mac, Windows, or Linux as well. And the good thing is that it's hardwired to the PDB. So if I want to download that PDB, which I saw before, a few pages before Disprot, yeah, this one, then you only have to copy the ID, which is 1 pkg in this case, and say file fetch by ID. Of course, it's under my chat window, but yeah, there are many different databases here, but the PDB is the default, and you just copy it here and push fetch, and it will come down soon. And you have the structure here, so you can rotate, you can color, you can highlight parts, so you can choose the sequence panel, which is there are 2 chains in this structure, 2 different proteins probably, or 2 different domains. If you just look at chain 1, I hope you can see my multiple windows. So here you already have some colorings based on the secondary structure element. I think the green ones are beta sheets probably, and the yellow ones are helices. But if you just simply drag and highlight this part, it will be also highlighted on your structure. As you can see here, it is now just highlighted by this green border. But if you want, you can, because for example, this is your part of interest, then you can just simply color it to red. And from that moment, you will have that part of your interest with highlighted by red. So there are a lot of different functions. Again, if you are interested, I'm very happy to show you all the parts. It can do some basic molecular dynamic simulations as well, but also a lot of different structure analysis. So I'm sure that we don't have the RMSD here, but I'm sure we have, for example, radius of gyration somewhere here, distances, angles, torsions. Yeah, here we have the structural comparison where we can have some RMSD as well. So that's one of my favorite programs to visualize a 3D structure, where you can also measure distances. You can also analyze bonds, but there are also many other things. The next thing, what you can just use as a snapshot of your structure and have some very basic understanding on the structure, is the 2D contact map. Let me just Google one for you. So it looks like this, for example. Let's see if we can see the full picture. So in the 2D contact map, what you define is that which amino acids are close to which other amino acid. And it's not loading in, but there was one, which I think it's the same, I think. Yeah, PLOS journal. Is it the same PLOS one? The same slow. Okay, we have the paper here. There will be a nicer one than this one. Down below, not that one, not this one. Yeah, this is it. So on the X and on the Y axis, no, on the X and on the Y, you have the amino acid position. So this is the 50th amino acid in your sequence. And what you see here, for example, what you see in the middle, of course, you will always have a diagonal, because neighbor amino acids are always close to each other, just because they are connected to each other with a peptide bond. But so that's why you always have a diagonal line. But for example, what you see here, if you see a widening, that means this is an alpha helix. So if you think about the alpha helix is a spiral where more amino acids will be close to each other. If you think about just looking at the structure here, if you have an amino acid here and another amino acid somewhere here, they are, in terms of position, this is 681, this is 685, so they are 4 amino acids away from each other, which is quite a lot of distance. They wouldn't be plotted as a close amino acid, but because they are close in terms of space, not that easy to move, they will have this specific pattern on the 2D plot. All the other ones here, these are all beta sheets, anti-parallel beta sheets, something like this here, this one, these two, sorry, this beta sheet, which turns back, and this beta sheet here. That's why you can, that's when you can see something like this. So that's again something which is a good thing to see, and especially if you see changes, then you can easily see changes on these 2D maps. The accessible surface and hydrophobic area, I spoke about that. It's very important and it's very easy to calculate. Programs, you can visualize the surface charge distribution. For example, by Chimera, you can, instead of visualizing the backbone, you can put the surface on it. I don't want to do that because it's sometimes a bit slow and I don't want to spend time with that, but it's you can color the surface by charge, and you can also see specific things on the surface. You can find surface loops. They can be very important. So if, for example, if you are targeting a globular protein with an antibody, then it's usually good to look for surface loops. So these are longer linear parts that are always exposed for the solvent and always exposed to other proteins. So a very good target for antibody epitope. You can see them sometimes from the Pdb file, but also if you just look at the visual inspection on this domain. Speaking about something like this here, this is a surface loop. It is visible clearly on the surface, and it's a long region. Or if you look at this loop here, if you have some kind of specific amino acid sequence on this loop that you can target with an antibody, it will be always accessible for the antibody, much more than something which is, for example, down in the middle of your domain. Okay, so that's something again what you can do, and also you can find contacts even with Chimera. You can define distances, and you can find hydrogen bonds. You can find hydrophobic interactions, for example, with Chimera again, which can help you to decide if an amino acid is important or not. If you see that an amino acid side chain is just outside of on the surface and doesn't have too many interactions, then probably it doesn't have too much effect on the structural stability of the protein. If you have a hydrophobic residue inside and you see 4 different hydrophobic interactions with other 4 amino acids, then it is probably a very important side chain in terms of stabilizing the protein structure. And now I will speak very shortly about molecular dynamic simulations. But again, if you are interested in molecular dynamic simulations, I'm very happy to help. The whole idea with molecular dynamic simulations is to simulate what is happening in real life. In real life, these proteins doesn't exist like this in the cell. They are always like wobbling, they are deforming, they are much more a dynamic thing and not a static, rigid structure. So what you do is that you take one molecule, one Pdb file, which, oh, I haven't shown you a Pdb file yet. Let me just show you one. It's ugly, but just to give you the idea what is the file you will start with. If you are on the Pdb website, you can say display Pdb format. That's the basic standard format for Pdb. It looks ugly. There is a lot of metadata and information here, which you will never use. But what I actually use is all the atom coordinates, which are starting somewhere down here, not here, here, at this point. So each line is defining one atom. First one has the serial number 1. So this is the first atom, which is a nitrogen, which is sitting in an asparagine. This is the 1-letter code. No, sorry, this is the chain identifier. So it's in chain A. This is the amino acid position within the original protein. And these are the XYZ coordinates because it's a 3D space. And there are some other information here. These are the most important information. So based on this set of coordinates, the program will build up this nice three-dimensional structure. So, and this is what you put into the molecular dynamics simulations. You put in this set of coordinates, and it will give an initial kick for the whole molecule. It will provide some kind of energy, some kind of temperature box. If you do it with a solvent, you can also put in water molecules into your simulation box, and then just leave it like that and watch what is happening. The ideal situation would be just to leave it on a room or body temperature and see how the protein is changing, because that's what is happening inside our cells. The problem is that we don't have the computational capacity to wait until it simulates minutes or seconds or hours. The recent simulation times especially for a domain are rarely more than pico or nanoseconds. So, and it takes days or weeks to simulate these. So if you want to do the simulation for longer time, or if you want to make a simulation on a bigger molecule, you have to do some kind of compromise, and you have to decrease your reliability in a way. The two main things you can do is, one is the coarse-grained simulation, when you, instead of simulating all the atoms, you are actually making beads. So you will have a bead for glycine, you will have a different bead for alanine, and you have 20 different beads with 20 different properties, and you are just looking at the interaction between these beads. It's a way much simplified way. The computer has to do way much less calculations, so you can simulate longer times, but you already put in a huge bias. The other way, which I like a bit more, is the discrete molecular dynamics simulations. So for that, let me just show you one picture. The only picture you have to understand is not here. I saw it today. Okay, let's just have this one, and then I will show you another one in another window. This one. So if you think about the interaction potential between two atoms, if we are speaking about, two or it can be like two hydrophobic side chains. It looks something like this when you have the distance on the x and you have the energy on the y, or the potential energy, and in the minimum, that's the point, that's the distance where the interaction is the highest, the best. So that's the optimal distance between the two atoms or two side chains. If they are getting closer to each other, of course, they will repulse each other. If they go further away from each other, then the whole force that are keeping them together will be weakened. So all of the forces between atoms, between side chains, between a negative and positively charged entity can be described by this kind of Lennard-Jones potential curve. The problem with the molecular dynamics simulation is that in each steps, in each attoseconds, or I don't know, in each very small time steps, when you do the simulation, you have to recalculate all the energies within the molecule. By looking at all the bonds, all the interactions, you will have to recalculate, okay, the distance is now moved from here to here, so the energy, potential energy, will go from here up to there. And this is what makes the whole molecular dynamics simulations extremely long, because you have to recalculate these potential energies every time during the simulation. In the discrete molecular dynamics, what you do is that you are simplifying this interaction potential curve. So instead of decreasing the interactions, what you do with the coarse grain. So if you think about the coarse grain, what you do is just exclude a lot of extra interactions, because instead of having tens of 20s or hundreds of interactions per one side chain will have only one with another one. Not all the carbons and all the oxygens or nitrogens will be interact with other atoms. That's the coarse grain. But in the discrete molecular dynamics, what you do is that you simplify this curve, and this is how it looked like. It will look like something like this. Sorry, it's a bit small, but oh yeah, this is the one I looked for. So this is the original curve, and this is the discrete curve. And the good thing is that you only have to recalculate your potential energy if it is stepping up from here to here. But as you can see, there is quite a lot of distance covered by the same energy level. Again, this is a huge simplification, but it will speed up your simulation a lot. Again, there are many variables you can introduce. Of course, there are different times you can run your simulations. The temperature is very important. You can do a simulation on a steady temperature, or you can increase the temperature if you want, and you can see how the molecule unfolds by temperature denaturation. You can also use different solvents, but this is a little bit artificial, to be honest. As I told you before, simulating folding with molecular dynamics simulation is very hard, because it's very easy to stuck into a local minimum. And that's why there is a special way of doing molecular dynamics simulations. And this is especially a very useful tool if you want to see structural variants for the same protein sequence, which is called replica exchange simulation. I will tell you in just one word how it looks like. It's actually quite simple. So instead of having one molecule in one environment, you are starting with multiple molecules in multiple environments. And the multiple environment is usually just simply multiple temperatures. So in this figure, what you see is that on the left-hand side, it starts with T1, T2, T3, and T4. Usually the T1 is a bit cold. T2, for example, should be a body temperature, and T3 and T4 are some higher temperatures. And what is happening is that you are running your simulations within these boxes, but in certain times you are allowing an exchange. This is why it is called replica exchange simulation, because you are multiple replicas and you are exchanging them. So as you can see here in the first exchange, the black and the red remained in its original temperature and environment and box, but the blue and green changed. So what is happening here? The blue was in a very high temperature, so it was probably a bit unfolded, but after the exchange, it went down to a lower temperature environment, which means that it will start to fold up again. And in the next step, it will go down even more. And in the next step, it will go down even more again. So this is something what will make the blue structure more and more compact, more and more going down to the folding state. But usually what you do is not exchanging 4 times, but you are running this simulation and exchanging them like 1,000 times. So all of these different replicas can walk through the whole, all the different temperature boxes, which is usually not just 4, but at least 8 or 12 temperature boxes. And they have the chance to go up a bit higher energy level and go down to a lower energy level. So what will happen with those folding funnels we saw before, even if they are falling into a local minimum, they will have chance to go up to a higher energy level and come out from that minimum, and they will have a chance to go down to a normal path. So you are actually sampling the whole folding space much better with replica exchange simulation. It's a bit longer, but it's not extremely long. We have about 20 minutes left, or 15. So if you have to analyze the transmembrane protein structures, I already told you about HMM top and CC top. The first thing is identifying if it is a TM protein or not. As I told you, you can easily misidentify globular protein as a transmembrane protein if you just put it into a transmembrane topology predictor. There are 2 things you can use. There is a Signal IP website, which is a well-maintained server. Signal IP. This is it. It is working for archaea and bacteria as well. Put in your sequence and it will try to find the signal peptide, which is responsible for targeting your protein to membrane. It's usually on the N-terminal. But in the same way, you can use the CC top. So if we go back to CC top here, and we go on the first side, so you can analyze your sequences by clicking here. And here you have to check in the TM filter and then put the sequence here. And probably if you put in a globular protein, then it will tell you it's a globular much more than a transmembrane protein. But I have to say that the first line of this slide is not 100% reliable. You can misidentify a globular protein even nowadays to a transmembrane protein. But if you are sure that it's a transmembrane protein, then identifying the topology is a bit easier. Maybe it is already in the TopDB, which I showed you. That was the one which the topology database, TopDB, this one. So you can here search for protein names or different identifiers, and you may have a chance that you already see topology within the database. If not, then you can predict the topology with the HMM top or the CC top. And also, yeah, that's the next step. If you have a topology, you may want to identify the sites. Sometimes there are different charge distributions. I don't know all of the different membranes and different sites, but if you are interested in this more, I can look after it and help. Which one is a bit more negatively charged, which one's a bit more positively charged. The reason is, again, we have so many different membranes and I don't know all of them. But there is also another tool called TopDom, this one. Again, a familiar name here, but this actually, this database is automatically extract domains from different databases, basically from Uniprot, that are sitting on transmembrane proteins, but sitting on one of the sites only. So if you can identify a domain or a short sequence that is always extracellular, then and you have it on your protein, then it maybe helps you to identify the outside and the inside of your protein. Of course, if you have that kind of transmembrane protein where you have a large extracellular intercellular part, you probably have to put that into PFEM to find the globular domains. And if you have much more that kind of protein, what we saw with the MDR, with this one, where you have less, much smaller loops here and there, then you have a chance to identify these loops and use these loops by looking at the disorder pattern, especially short disorder pattern on these regions, and try to find out something more by that. And finally, I think this is, yeah, finally, if you have a disordered region, first you want to identify again, which can be done by IUPRED. So if we put in a real disordered protein here, for example, the P53, and Uniprot is not extremely fast at the moment. It's here, TP53, and I just copy the sequence. And put it here. When I spoke about long and short disorder, here you can choose short disorder. So if you have a transmembrane protein and short loops, then you may use the short disorder prediction. But here we use the default, which is the long. And as you can see here with the red, you can identify the disordered regions quite easily. Everything that is well above the 0.5 has a very high probability of being disordered. The blue line is actually an extra information that's also here on my second line. So if you have a disordered region, you can have you can use NCORE, which is the blue line, to highlight the potential interaction regions. So for example, within this quite long disordered region, there is a high propensity interacting part. This is usually just a small 8-10 amino acid long region, which has a very high tendency for making protein-protein interactions. So it's again a very useful information for you, because maybe this is the region what you are looking for to inhibit if you want to inhibit a protein-protein interaction. You can predict secondary structure by using these programs. I don't really like using them. I don't think they are extremely reliable, but you can. These are just based on the sequence. If I really want to see the secondary structure propensity, which is actually extremely important here, and this will be my final message for you today, then I'm using molecular dynamics simulations, especially replica exchange molecular dynamics. Usually you only simulate a very short region here, like 20, 30 amino acids. It's very fast. It's less than a day to do molecular dynamic simulations on a region like this. The reason why it is extremely important is that because disordered protein regions can interact with other proteins in two ways. The two main way they are interacting is conformational selection and induced folding. Induced folding is the rarest one and the simplest one. So that's why I will say this first. So what is happening with induced folding is that there is a special, let's say, secondary structure, let's say an alpha helix on the disordered part that is capable of interacting with the interaction partner. But this alpha helix is not there. It is going into an alpha helix when it is partially interacting. So there is a first interaction governed by only a few amino acids between the two proteins, but then protein A, which is probably a globular protein with a large surface, will induce the folding of the disordered part, and it will take its final interacting structure by the help of the interaction partner. In this case, it is not really important what was the secondary structure, because the secondary structure, the binding secondary structure form, is always induced by the partner. But in most of the cases, this is not the situation. The situation is that the interaction partner can only interact with one specific structural form of the disordered protein. So if it's an alpha helix, then it can interact with partner A, but if it's not an alpha helix, but it's a more stretched region, it can interact with partner B. And of course, if this whole interaction is sensitive to the concentration, then you actually don't really think about the concentration of the protein, but you are thinking about the concentration of that structural form that is capable of interacting with the other partner. So if you have an alpha helix tendency within a region, and this tendency is 60%, then only 60% of all of your proteins are capable of interacting at one time with the partner, or there is a 60% probability that your disordered region will be in that form, which is capable of interacting with the other protein. This maybe sounds as just like a minor thing, but to be honest, this is extremely important in disordered protein interaction. Let me show you a paper about this. This was actually published in Nature, so no, Nature Chemical Biology, sorry. This is one of the most important interactions in the human cell, p53 MDM2 interaction. This is how the p53 future will be decided. If they are interacting, then the p53 will be degraded. But the most important thing here is that this is, again, a good example for conformational selection. So if you look at this picture, MDM2 is only interacting with the alpha helical form of p53 within this region. This is a quite small region, very short region, a few like 5 amino acids, 6. Yeah, so the red part here on the right-hand side, these 6 amino acids are responsible for the binding only. And what they did, they replaced a few prolines outside of this region. So these prolines here and here, they are not responsible for binding. But if you replace them, then you will shift the alpha helical propensity. If you can see here, the helical propensity or helicity here is around 20–30%, while here it is going up to 60%. And here, if they change the first 2 prolines as well, it goes up even higher, more than 60%. So what they did, they made p53, the region of p53, more helical, which means that it will bind to MDM2 more often. And this has actually physiological consequences, just increasing from 30% to 60%. It actually means that you are doubling the helical propensity. You are doubling the amount of short regions that are ready to interact with MDM2. So it's like doubling the amount of protein in terms of the interaction here. So you are actually shifting the equilibrium with this. So yeah, this is just my last point here. So if you encounter with disordered regions, and there is a chance that even prokaryotes, encounter, then it's much more about the local structural propensities, the local amino acid changes. When I told you about few hydrophobic residues. So if you look at this sequence, this is very hydrophilic. These are all hydrophilic residues here, but you have a few hydrophobic ones, and they are big ones. It's a phenylalanine and a tryptophan here, and they are usually having some kind of very important function. So, but again, this is you have to think about very small things in disordered regions. So I think that's the end today. If you want a bit more about structural biology, I'm very happy to do another meeting where we only do practices and we look for sequences, we look for how you can process the Pdb file, how you can do some very basic structure analysis in Chimera, for example. But I hope that this one was at least enough to show you the problems you can encounter when you do structural biology. So I think I will stop sharing here, and if you have any questions, I'm very happy to answer.